So our next speaker is Dr. Alex Harari, who joins us from the Ludwig Institute in Lausanne. Dr. Harari was an accomplished traditional immunologist, I'd call that, in 2014 already when he joined the institute, which is led by Dr. George Koukos. And earlier this year, I had the privilege of listening to Dr. Koukos' talk, and he described how even the architecture of the institute is actually laid out in a way where you have all the disciplines coming together for harnessing immuno-oncology for cancer therapy. And I thought that was a very exciting thought because this convolution or this coming together of convergence of fields is something that all of us, of course, are excited about. So I'm very curious to hear you describe your work in that environment. Thank you so much.
So thank you for the introduction and the invitation. So indeed, I will only present an aspect of all the research done in Lausanne, and mainly the T-cell profiling aspect. So as I said, I will talk about T-cells, and in particular Neoepitope reactive T-cells, which since a couple of years are getting very attractive. So as you all know, there are different classes of tumor antigens, shared tumor-associated antigens, and neoantigens. The one I will refer to are in this study are exclusively derived from non-synonymous somatic mutations, which accumulate in the tumor. So these are private epitopes. And what made the tumor neoantigen very attractive, at least from a theoretical standpoint, is that by definition the tumor neoantigens are exclusively tumor-specific. They are not expressed in non-tumor tissue. And also, because they are non-self, the T cells recognizing this epitope should not be affected by mechanisms of central tolerance. So from this, there's been an overwhelming number of reviews and so on, papers showing how neoepitopes could be exploited for therapeutic purposes. For instance, here this very good review by Ton Schumacher and Bob Schreiber, showing, for instance, that you can identify neoepitopes in the patients and then design synthetic vaccines with these epitopes to immunize the patients. This has been done in some tumor indications with very amazing clinical data. Another opportunity is to identify neoepitope reactive T cells, purify these cells, expand these cells, and reinfuse into the patients. This also has been done in very isolated cases with very amazing clinical response. So there has been, in the meantime, overwhelming evidence that indeed these neoantigens are associated to the efficacy of immunotherapy, either as a biomarker for checkpoint blockade inhibitors, or, as I said, for also adoptive cell transfer therapy. But most of these studies were performed in high mutation load patients. While still in the same review I mentioned, the authors upfront raised the point that maybe given the very wide heterogeneity in the tumor mutational burden, so the number of mutations per patient, and which is different among the different tumor indications, that maybe some patients would not be eligible for these mutanome-based immunotherapies. Hence, they artificially stratified basically all the indications with somehow the likelihood to identify new epitopes. So the first question we asked was indeed whether all patients would be eligible for mutanome-based immunotherapy. And so in practice, the question was to see whether we can identify new epitopes in low mutational load patients. And then another question was whether we can induce de novo, so without any synthetic vaccine, whether we can induce de novo T-cell response against neoepitopes in agnostic way. The following question was to see whether neoepitope reactive T cells are somehow identical or equal in terms of functional profile. And so the question was also then if there is functional heterogeneity among these cells. And guessing that there is in fact functional heterogeneity, then what would be the best parameter, or the best functional parameter that would correlate with clinical efficacy? What would be the best immune correlates of efficacy and the most clinically relevant functional parameter? So these are the different topics I will try to cover. So I start with the identification of neoepitopes in low mutational load patients. So I focused here on a cohort of more than 40 patients. 25 had advanced epithelial ovarian cancer, and most of the data I will show you are on these patients. And then we have more than 15 other patients with lung, GI, and melanoma cancer, which I will also discuss. What is important is that all the patients that I will show you the data on were naive to immunotherapy. So I start with the ovarian cancer patients, and this is just to show you where ovarian cancer is in this spectrum of tumor indications. So it's really a prototypic example of low to moderate mutation load patients. The strategy we follow to identify these neoepitopes is very consensual. We used, I won't go into detail, we used public algorithms to predict class 1 neoepitopes, and we had usually about 100 peptides, candidate peptide, to interrogate the immunity of the patients. So we started the analysis on the blood. This is the work of Sarah Bobis, and the paper has been published earlier this year. So she basically interrogated the circulating T cells from the patients and successfully identified neoepitopes. So you have here one typical example of validation of one neoepitope with the detection of the mutated peptides and lack of reactivity against the wild-type counterpart of the epitope. And whenever this was possible, we validated then the reactivity with peptide MHC multimer complexes. So we successfully identified neoepitopes in about 1/3 of the patients when we interrogated the blood. Of note, in collaboration with Professor Ferrer, also in Lausanne, he looked at the gene signature of the tumor among the patients with spontaneous detection of neoepitopes in the blood versus those with undetectable neoepitopes. And indeed, he found very nice associations with basically 3 key signatures: antigen processing and cross-presentation, PD-1 signaling, and interferon gamma signaling. So the tumors from the patient with detectable neoepitopes in the blood had these enriched gene signatures. We then moved forward and interrogated the TILs, so the tumor-infiltrating lymphocytes, from the same patients. And the TILs were expanded using conventional protocol, which is basically high-dose IL-2. And so when we interrogated the TILs of the patients, again, we successfully found a few neoepitopes. Also in the same range of patient, between 1/3 and 1/4 of the patients had neoepitope-reactive T cells found in the TILs. But then surprisingly, when we compared the repertoire of neoepitopes between the blood and the TILs from the patient, so each column here is a patient and a color square is a neoepitope, we found that basically the repertoire were quite discordant. So with only one exception here in yellow, in most of the cases, the neoepitope-reactive T cells were either identified in the blood or in the TIL, but in most of the cases, not in the same patients. So this was this came as a surprise, and so we challenged basically this observation. We, for instance, took here these patients where the neoepitope-reactive T cells were exclusively detected in the TIL. We managed to purify, sorry, the definition is not very good. So we purified the neoepitope-reactive TILs, analyzed the TCR repertoire, not with the same expensive company discussed in the previous talk. This is an in-house assay that we have, and then looked for the dominant TCR within the repertoire of the different compartments. So we first looked at total TIL, and actually indeed confirmed the presence of this TCR among the TILs. The TCR was also present among the T cells from the primary tumor, but we failed to detect it in the peripheral blood, so it was below our threshold of detection. So this was very consistent with our screening, with our functional assay. We then did the reverse exercise. We took one case where the epitope was exclusively detected in the blood. Again, we sorted the cells, analyzed the TCR, looked for this TCR in the peripheral blood where we found it. Of interest, we found this TCR in the primary tumor, but not in the expanded TILs. So, suggesting that indeed the clones were present in the tumor, but failed to expand during the TIL expansion protocol. So this, and I think many others made the same observation, and this also combined to the fact that in contrast to the blood, there was no association between T reactivity and any gene signature in the tumor, basically suggested that for some reason the way TILs are expanded is suboptimal and that there's room for improvement. And so we made a very simple hypothesis, which was that we could improve TIL expansion protocol by simply adding the antigen, so adding the predicted peptide. And so we did the experiments comparing the conventional TIL expansion, so that is in practice to take the tumor cells and to either put high-dose IL-2 during a couple of weeks, or we tested what we call the prime protocol, which is to add, in addition to high-dose IL-2, the pool of predicted peptide. And this indeed led to very nice data where clearly we could see significant enrichment in the frequency of neoepitope reactive T cells in the TIL with our prime protocol as opposed to conventional, so that's a 30-fold enrichment. But we also saw the case where there was undetectable response in the conventional TIL, but we could detect a very clear response in the primed TILs. And here are the cumulative data showing that indeed we could either enrich for pre-existing, if you want, epitopes, or unleash the presence of new epitopes in the TILs. And so with this little trick, then the proportion of patients with detectable neoepitopes in the TIL shifted from 28% to 75%. And when we combine the detection in the TIL to that of the PBL, then we could detect neoepitope reactive T cells in mostly in the majority, almost all the patients. But still, the repertoire between the blood and the TILs remains mostly discordant. We had only 3 common epitopes recognized between the PBL and the TIL. So before I move to the functional profiling of these cells, I just want to briefly mention that, briefly also because this had also been published earlier this year, that these patients with ovarian cancer were enrolled in a vaccine trial, in a personalized vaccine trial, where the patients were immunized with monocyte-derived dendritic cells, which were pulsed with oxidized whole tumor lysates. And the patients received multiple immunization with this personalized vaccine. And the trial was performed at UPenn, but all the analyses were performed by us in Lausanne. So very briefly, the vaccine was immunogenic. So in majority of the patients, we could see increased frequency of T cells recognizing the vaccine. And this increased responsiveness against the vaccine was associated to improved survival. And the improved survival was also stronger in the patients with increased reactivity against the autologous tumor. But then, of course, we also analyzed the neoantigen landscape in these patients. So what we saw, you have here 2 representative examples, was that, for instance, in these patients, there is a very clear neoepitope recognized only after vaccination, undetectable before vaccination. This is also the case for this epitope in the second patient. And in some cases, there was what we saw in 1/3 of the patient, pre-existing neoepitope activities massively expanded by vaccine. So overall, we saw in the majority of the patients either increased frequency of neoepitope activity after vaccination or the de novo detection of new neoepitopes, showing that although this is a personalized vaccine because it uses the autologous tumor, this is an agnostic way to induce neoepitope activity in patients with low mutation load. So having accumulated so many, more than 40, I believe, neoepitope clones, we then performed a deep functional profiling of these cells. We first started with basic intracellular cytokine staining, evaluating the polyfunctionality of the T cells isolated from the blood or the TIL. This is a somehow outdated assay, but it was BD, I remember, played a very pioneer role in the development of this assay 15 years ago with Holton Maker and Skip Mino and all these people. But okay, it's outdated, but still, I used to do that, so I did it again. So we assessed the functional profile of these T cells, and we could see that in both the PBL and the TIL, most of the neoepitope-reactive T cells are composed by polyfunctional T cells. We then made a deeper analysis, focusing here on one epitope, neoepitope, recognized by the PBL and the TIL. So we'll first managed to purify and then expand neoepitope-reactive PBL and TIL, looked at their TCR repertoire, and could see that despite the fact that the cells recognized the same epitope at the same time point in the same patient, the repertoire were different. So there were not the same clones identified in the TIL and in the blood. When we did, by site of a deep functional profiling, so we repeated an extended intracellular cytokine staining with polyclonal stimulation and could see that the TIL and the PBL were very similar. We did a phenotype, and again, both, you have here the pair of PBL, pair of TIL, very similar phenotype. So the cells look quite similar when we analyzed this way. We then performed a deeper assay assessing the functional ability of the T cells. So this is the antigen sensitivity. It reflects how prone are T cells to respond when they are exposed to low dose of antigen. And so when we analyzed here the pair of PBL, the different clones obtained from the blood or the TIL, again recognizing the same neoepitope, we could see here that the TIL had a higher functional ability. This was true for a couple of cytokines, but also for the killing ability. And this was also associated with improved signaling, again performed by CyTOF, from in the TIL as opposed to the PBL. We also had the opportunity to collaborate with Professor Zoet, who is a bioinformatician from Ludwig in Lausanne, and so he did molecular modeling using the sequences we had obtained, the TCR sequences. And so basically he estimated the interaction between the peptide MHC, which is common here between the PBL and the TIL, and the different TCR, and he could see that indeed TIL were equipped with TCR making more favorable interactions with the peptide MHC, so very consistently with our functional assay. And then we extended all the analysis to all the different neoepitopes that we had identified in the blood and the TIL, and again confirmed that although there is a clear overlap, the TIL tend to have a higher functionality as opposed to the PBL. But still, this is a relative comparison, so we wanted also to compare from a more absolute standpoint with other classes of antigen. So, okay, work in progress, we need to understand that. We then moved to another assay, which is measuring the TCR ability, or the structural affinity, which measures the affinity of the TCR for the peptide MHC. So that's a biochemical assay as opposed to the functional ability, which is where the readout is the ability of the cells to respond. So to measure the structural affinity, we use a tool developed by Julien Schmidt a couple of years ago in Lausanne. So this is a dual color peptide MHC multimer, and this is reversible. So each monomer, so peptide MHC monomer, is associated to a dye, which is Cy5, and then the multimer is associated to phycoerythrin, PE. So you can use as a conventional tetramer or peptide MHC multimer, but then upon addition of imidazole, there is dissociation of the complex, and then you are left with the monomers bound to the TCR. Since each monomer has its own dye, you can follow the dissociation of the monomer from the cell and follow this by the loss of fluorescence in this dye, which is Cy5. So the half-life of decay here is associated to the off rate, so the affinity of the peptide MHC, of the TCR, sorry, for the peptide MHC. And so what are the consequences of this functional heterogeneity? I don't have a clear answer. I think more work is needed to understand also which parameter is the best predictor of clinical efficacy. So in conclusion, I showed you, and this was also confirmed by others in other tumor indications. So we focused here on ovarian cancer. The recognition of neoantigens occurs at a high rate, even in low mutation load cancer types. The way TILs are expanded is suboptimal, and it can be optimized to favor to enriched in neoepitope reactive T cells. Neoepitope reactive T cells from the blood and from the tumor are functionally heterogeneous, in particular with regard to the functional avidity, but also the structural affinity. We have observed, or we have preliminary data suggesting that the functional avidity does correlate with the intratumoral frequency. And so overall, this calls for the need of new tools to make high throughput, not only screening of TILs and blood, but also new tools to filter among the different neoepitope reactive T cells, which are the fittest, and so which one should be prioritized for personalized immunotherapy. So I think I mentioned most of the key collaborators. Again, I'm working in the group of George Kukos in Lausanne. I thank you for your attention, and I'm happy to answer any question.